Imagine a construction site, but on a molecular scale. You are watching DNA being copied, and the stars of the show are a family of specialized enzymes called DNA polymerase. Nature's microscopic builders. DNA polymerase can only build in one direction from 5' to 3', but DNA is double-stranded and anti-parallel. So how do we copy both strands? The leading strands run 3' to 5' towards the 4, so the polymerase can just glide along. Easy and continuous copying occurs in the leading strand. Second is lagging strand. Lagging strand run 5' to 3' towards the 4. That's backward for the polymerase. Copy it in short chunks called Okazaki fragments. Each builds 5' to 3', then glue together, later by DNA ligase. The primers. DNA polymerase can start from scratch. They need a free 3' end to get going. So the cell uses an enzyme called primase to lay down short RNA primers on both strands. From there, polymerase takes over building of that starting point. Now the DNA replication. To make sense of it all, let's break the process initiation, where it all begins. Replication does not just start anywhere, it begins at specific launch sites called origin of replications. In bacteria like E. coli, there's just one origin, known as ori sites, a 245 base pair sequence that attract the replication crew. But in complex organisms like us, human cells use over 20,000 origins to copy their enormous genomes quickly and efficiently. Helicase. Think of it as the unzipping enzyme. It pulls the two strands apart. Single-stranded binding proteins. These keeps the strands from snapping back together. Topoisomerase, like DNA gyrase, they relieve the tension that builds up ahead of the unwinding helix, like unscrewing a twisted rope. But wait, DNA polymerase, our master builder, can start construction without a foundation. That's where primase comes in. The primase lay down a short RNA primer 5 to 15 nucleotides long, giving DNA polymerase a free 3' end to work with. Only now can replication truly begin. And when it does, the real action starts. Replication does not just move in one direction, it moves both ways from each origin, forming two replication forms. Here's the twist. DNA polymerase only binds from 5' to 3', but DNA is anti-parallel, meaning the two strands run in opposite direction. So how do we copy both strands? Leading strands. Easy. DNA polymerase slides smoothly along, building continuously. Lagging strands. Not so easy. It has to be copied backwards in short bursts called Okazaki fragments, about 1000 to 2000 bases each. To do this, the lagging strands form a loop, flipping orientation just along. just long enough for the polymerase to build a new fragment. Once it hits the previous fragment, it detaches, form a new loop and start again. This gap between Okazaki fragments are sealed by DNA ligase, linking the sugar phosphate backbone. DNA polymerase 5' to 3' exonuclease activity helps clean up leftover RNA primers. Together, it's a seamless relay race of fragment building and joining, all happening in milliseconds. We have studied the different processes of DNA replication: initiation, elongation, termination.